hydrophilic section. That's for the carboxyl. Okay, because carboxyl is a little bit different. That's why all these three together we call it a parallel casing. There is a tons of research did in China before. When we eat casing, the casing go to the stomach. We have a stomach pepsin. The pepsin can hydrolyze casing and will generate some of the antimicrobial peptides. Have some antimicrobial function. Okay, so this is what I want to mention about the casing right here. The next one I want to briefly mention about the lactoglobulin. Okay, lactoglobulin, I want to go up right here. Number one, beta-lactoglobulin is rich in lysine, leucine, glutamic acid, and aspartic acids. Their components are rich in these 4 amino acids. Number 2, there is AB2 genetic variant there, major AB2 variants. The molecular weight is about 18K delta. And very important, this A variant contribute to a very interesting cooked milk flavor. And the beta-lactoglobulin is less soluble, very much calcium sensitive. Okay. How about lactalbumin? Albumin, I don't want to mention too much. It's not variable. It's just you have egg albumin, everything there. It's a single protein, simple protein. Alpha-lactalbumin, you also have the AB2 variants. Molecular weight about 14,000 kDa. Sorry, 14 kDa. So not really pay much attention to forming of the gel because it's small. This guy, you know about it, is that right? Milk products have some IgG, IgA, IgM. What does the immunoglobulin looks like? What's the IgG structure? We talk about this in my class, other classes. We talk about it in general microbiology class too. What does it look like? SS double bonds, is that right? What is this chain? H chain. Heavy chain. What is that one? Light chain. How are they connected? With disulfur bond. What at the top here? Variable ends. How about here? FC section, is that right? The variable ends will be connected with antigen. And this is the antigen. Connected. How about the FC? What is that? Macrophage. The terminology called opsonization. Is that right? How many amino acids in the heavy chain? Like 50,000. How about the light chain? Maybe 25,000, 20,000. That's another class. We don't want to talk about it for the microimmunology class. Is that right? So this is what we talk about is a milk protein. And very interesting, lots of information there. Our textbook actually wrote, I will say it's a little bit messed up there. Somewhere here and there, you can see it. Very brief, but it's kind of confusing you, but so I want to lay out that. Now the very important is their components is important. The casing, the lots of things we can talk, and how you're going to do a brief isolation. The detailed isolation you have to use the other methods, like G16, Cepadex, column, all those things. And that's what we did a long time ago. People, dairy science is interested about all the components of these milk products and the proteins in the milk products. Okay, so that's all I have today.